All right. Hi, everyone. Welcome to the Harlow seminar series for tonight. As some of you know, if you read our last minute email, we're having a little bit of a seminar surprise this evening. Bob Smith, our intended speaker, let us know this afternoon that he wasn't able to make it due to a medical emergency. So we brought in the A team, the four gentlemen in the front row here, who, as of 1 o'clock, have prepared a seminar for you. So, round of applause. So my name is Sarah Collins. I'm the station director, and I'm also part of the project that they'll be talking about is called WyACT. I'll explain what that is in a second here. And so the general topic of the four presentations are all about the ecology of Jackson Lake, right out the window here, and then also the Snake River. So what WyACT stands for is Wyoming Anticipating Climate Transitions. This is a $20 million grant to the University of Wyoming that's in its 4th out of 5 years. And the overall goal for the project right here is to predict ecological and societal consequences of climate change in the nation's critical headwater areas. So the Upper Snake River Basin, where we all are right now, is one of those critical areas, both for things like water quality in the picture on the left, habitat for important fisheries like the cutthroat trout on the bottom right. And this is a really interdisciplinary group. So we have folks who are running global climate models and downscaling them to areas like our region or Wyoming or this watershed. We've got watershed hydrologists and ecologists who are thinking about how much water is moving through the landscape. We have our team, the freshwater ecosystem group, thinking about lake and stream ecology. And then on the back end, we have social scientists thinking about how do people care about water? How do people care about climate change? How much are they willing to pay to have fisheries, clean water, other things like that under different climate scenarios? So like I mentioned, our group is concerned with freshwater ecosystems. That's me on the left. Lily Fetzer and Annika Walters are the other two faculty members who are doing freshwater ecosystem work here and working on the project. And then we have a technician and 4 graduate students. The 4 guys on the left will be giving the talks tonight. And Meredith is the other PhD student who's not here today. Who are doing a lot of the work with focused projects on either the lake or the river, different facets of that. And so overall, we've got a lot of different approaches for this. The left is a Snake River temperature network where we have temperature loggers out in different tributaries in the main stem of the river in collaboration with a lot of agencies here in Teton County. You can see a water sample in the middle for zooplankton there that somebody collected on the lake. And then we also have a lot of sensor infrastructure, like the picture on the right. If you ever see a little blinking light in the middle of the lake out in the middle of the night, that's our buoy. It collects water quality data once every 10 minutes. And Kevin will talk more about that. So for the next hour, I will get out of here quickly. We first got Sean and Tristan thinking about water level changes in Jackson Lake, how that affects water quality, productivity, and the food web. We'll have Kevin talking about using these sensor networks to monitor water quality trends and patterns over space and time. And then finally, we'll have Chuck talking about water temperature and trout habitat in the Snake River. So I will get out of here so they can get started. And first we have Sean and Tristan coming up for that joint presentation.
All right, yeah, thank you everyone for being here. My name is Tristan Blechinger. I'm a PhD student in Lily Fetzer's lab.
And my name is Sean Bertalot. I'm a PhD student in Sarah Collins' lab.
And we'll together spend the next 20 to 30 minutes or so talking about our research and what we're doing out here. Kind of this integrative approach between Sean's work and my work, really focusing on this integrative study of the aquatic ecosystem out here in Jackson Lake. And so just a quick little overview about what we're going to talk about. We'll go do a quick overview to introduce you guys to some of the topics and words that we'll be using to describe this kind of stuff. We'll do a quick overview of our work on mapping aquatic habitat zones. And then Sean will talk a little bit about his research on primary productivity in Jackson Lake here. And I'll talk for a little bit about food web dynamics and the fishery in Jackson Lake.
Sweet, yeah. So just to kind of get started here, taking a little bit of a step back, thinking about like why aquatic ecosystems, why lakes, and why rivers, and why are these important? In aquatic ecology, we really like to think about these systems as sentinels of change. They exist at the lowest point in the landscape, and if you think about that, anything that happens on the landscape is eventually going to drain into these lakes and impact things going on in there. So by studying them really closely, we can kind of get a better idea of how not just things within the lake are changing, but also how changes on the landscape and within the watershed are driving these changes. We tried not to have too jargony of a talk today, but there were a couple terms that we needed to get out of the way because they're very important to what they're doing. So here we kind of have our 2D visual of any lake, Jackson Lake, for example, and you can kind of divide this habitat into 3 different zones. First of all, you have the profundal zone. This is going to be this really deep, cold area of the lake. This is a spot where no solar radiation is reaching. It's going to be very cold. There's not going to be a whole lot of plant growth or algal growth down there. Maybe a couple fish, but really not all that relevant for the rest of the system as the other 2 areas. The next up, we have the area where the sun is penetrating the water column, but not actually reaching the bottom of the lake. This is called the pelagic zone. So this is going to be warmer water. This is where you're going to see all of your algal growth happening, a lot of fish living out there because they're eating the zooplankton that live there. So this is a very relevant part of a lake, especially a deep one like Jackson Lake that has a lot of area that's very deeply penetrated by the sun, but isn't necessarily reaching the bottom. Finally, we have the littoral zone. This is the area where the solar radiation is actually reaching the very bottom of the lake. And because of this, you can have plant growth there. You can see your macrophytes as well as benthic algae on rocks. If you ever go to any of the lakeshore here and see kind of that green scum growing on the rocks, that's going to be that algae. This provides food for bugs living down there, which is, of course, food for fish. So this is a very, very important area of the reservoir and something that both Tristan and I's work really focuses on in different aspects. And then the benthic area is going to be just talking about that very bottom part of that littoral zone. So why is the littoral zone important? Because it's the interface between terrestrial and aquatic ecosystems. Anything that runs off is going to be running into the littoral zone first before it reaches any other deeper reach of the lake. It's a hotspot for primary production, so that's all that photosynthesis, whether that's the plants, the algae growing on rocks, or the algae in the water column. And then it's also, because of that primary production, a hotspot of biodiversity. It's where all the bugs and fish and other things really like to group up and live in these areas. And when we think about a reservoir like Jackson Lake, we know that this is an artificial reservoir, there's a dam on it, and we have fluctuating water levels. Because of that, those fluctuating water levels can impact the extent of habitat depending on the slope of the lake, lake bed, and the amount of drawdown. So sometimes a little bit of drawdown might not actually affect those ratios all that much. A lot of drawdown might have much more detrimental or obvious effects to the lake ecosystem. On top of this, we're talking about light penetration in the water column. So if we get growth of algae or we get sediment coming in off the lake, that also reduces light penetration, so it kind of shrinks that habitat. And of course, these 2 things don't happen in a vacuum. You can have more sedimentation, but you can also have lower water levels, all of which can kind of affect these littoral areas in really profound ways.
So, taking that into consideration, when we put all this together, Sean and I are working together on this project to map these different aquatic habitat zones that he just talked about. And the question that's really driving this project is, how do these water level fluctuations and changing water clarity across the lake over time as well impact this really important littoral habitat? And so for Jackson Lake here as an example, last summer, we spent a lot of time driving around recording depth at a whole bunch of locations around the lake so that we could put together this really high resolution bathymetric map, basically showing us the shape of the lake. And we can pair that with this daily storage data or the height of the water from the Bureau of Rec and water clarity data that we collect through our routine monitoring out here. And what we can do is we can estimate where this littoral habitat is as the water levels go down. And so this is really starting to set up the foundation for both my work and Sean's work to where this primary production, this algae that's growing on the rocks and these plants and stuff, where that can actually grow in the lake. And of course, this has major implications for the fishery and the food web and total production within the system. And when we compare this from the full pool to maximum drawdown, which out here is 39 feet lower, we can see that we lose a little over a third of this littoral habitat. And it's important to consider, even though this littoral nearshore area is really important for the ecosystem, the fishery especially, the warm top layer, that epilimnion, and the cold bottom layer, that hypolimnion, those change too, right? Because Obviously, if we're going to take water out, the total volume of these habitats is going to go down. And so we see this warm layer is reducing by about 33% from full to maximum drawdown. And that cold water volume, again, like the fishery out here, right, is a lot of cold water species. So lake trout, cutthroat trout, things like that, mountain whitefish that really like that cold water. They're losing almost a quarter of that habitat. And so when we're looking at this, this is really kind of trying to lay the groundwork to understand how these habitat changes driven by these water level fluctuations might impact these higher trophic levels and production within the system. And so while we've done this for Jackson Lake, we're also actively working on doing this for a bunch of other reservoirs around the state. So Boysen on the top there and Flaming Gorge on the bottom are two of a handful of reservoirs that we're doing this for as well, with the ultimate goal of developing an interactive web tool so that managers or any member of the public interested in this kind of thing can go onto this website and see how these things might be changing throughout the course of the year and over time as these water levels fluctuate. And with that, I'll hand it over to Sean for his talk.
All right. Yeah. So Tristan and I are both working on this because what we just talked about kind of sets the physical foundation for both of our work. But then it kind of divides up a little bit where Tristan's going to be talking a lot about the fishery specifically and food webs and stuff like that. I'm a little more interested in the base production of the ecosystem. And all of that is talking about the photosynthesis of algae, both growing on rocks and in the water column, as well as plants. And of course, this is kind of the base food that eventually carries its energy up to these fisheries. So both of these are very interconnected, and Tristan and I are kind of looking at two pieces of a whole in the sense of our projects. But my project specifically is tracking this primary production and thinking about ecosystem metabolism between different habitats within this very large, very unique reservoir we have right outside our doorstep here. But kind of taking a step back here, what is aquatic ecosystem metabolism? Or what is it even ecosystem metabolism, and why do we care about that? So when we're thinking about these integrative approaches, thinking about how the landscape impacts things, aquatic ecosystem metabolism is a really powerful tool for us to better understand how our aquatic ecosystem responds to different climate drivers, whether that's temperature or precipitation or other kind of physical chemical factors within the water column. And at its most base form, what we're talking about here is how does the ecosystem breathe? So in the same way that you or I breathe in oxygen and respire CO2, the lake ecosystem does that same thing, but on an ecosystem scale. So we have all of our plants and algae and fish and bugs, and all of these are producing CO2 when they respire. But of course, some of them, like the plants and algae, are producing oxygen. And unlike on land, in a lake, this is a closed system. So it's a loop where we can track the changes in oxygen and CO2 and figure out how the ecosystem is actually responding in real time to seasonal temperatures and other drivers of change. And so when we put this all together, we get a really comprehensive view of what is important for this ecosystem in terms of what's driving the energy flow through the system. And this inherently links to the landscape because all of the food that gets into this, all the base nutrients, a lot of that is coming off the landscape, coming through snow melt in the spring. So it's really important to kind of track those changes. And then we can determine sources and sinks of CO2 because one area of the lake might be respiring a lot more CO2 than another area because of a whole bunch of different factors. And so the goal here is to quantify the area of littoral zone primary production to overall primary production. So we're talking about that nearshore area versus that really deep offshore area. And we're also trying to provide a baseline for primary productivity in Jackson Lake so future changes can actually be tracked. Because if we don't have that baseline, how can we know it's actually changing? And so my research approach, this was last summer. We had 7 buoys out there. They're these little red small ones compared to our big yellow one. Had some annoying lights on them that we took off this year. And they've got these really cool dissolved oxygen sensors on them. And when you pair those with all the information coming off our big yellow buoy, you can actually model that flow of carbon and oxygen throughout the entire year. So they're taking data at a 10-minute interval pretty much constantly. And then we went out every week to take samples of chlorophyll, which is algae, and then also total nitrogen and total phosphorus, as well as our water quality depth profiles. And when you put all of that together, you actually get to build out what is the base energy for your ecosystem and how it changes throughout the year. And today I'm just going to be talking about the data that is coming off of those sensors and how we modeled that and kind of the results of that. So here we have gross primary production. So this is the total energy production for the reservoir at these different sites. We had 4 shallow sites and 3 deep sites. And what was really interesting is early in the season, these blue lines, you can see that we have this big peak in approximately like June, July, as you're a little bit far away from the Snake River inlet, but not too far away. It starts to fall off after that. And then when we get later in the year, everything kind of flattens out. So what this implies is that that snowmelt pulse we get through the Snake River when discharge levels are really high, that's actually driving a lot of the gross primary production within this reservoir. And that snowmelt seasonality and how much is actually getting put into the rivers might be a huge factor for how primary production and how algal growth is actually happening in the lake. And we see this too when we divide it up into sections. So our most northmost section, closest to the snake, is actually very low productivity. But then when we get further away, when you start getting more water volume and all those nutrients kind of start circulating through, that's where we see that really big spike in primary production early in the year. And then by the time you get down towards the dam, that's where you might notice it might be a little bit clearer, relatively speaking. That's because we've kind of run out of food for the reservoir. So that central part is probably really important for the whole lake, whereas these other 2 parts might not be contributing quite as much. So for some of my future work, I'm kind of following up on a lot of this. I'm adding in some satellite remote sensing so that we can take a look at those couple reservoirs we did with littoral habitat, but expand that to a lot more for not just Wyoming, but the whole region. On top of that, we've added buoys back in Jackson Lake this year, as well as other reservoirs across Wyoming. So we kind of have a reference in Jackson Lake and compare that to very different systems across the whole state. And finally, the buoys out there this year are actually going to be year-round buoys. So they're going under the ice. They're going to be staying out all year round. Because intuitively, if that snow melt is really important, probably that short little ice melt phase when the lake breaks up is also very important. So we're hoping to track those changes through the winter and into the spring next year.
Cool. Yeah. So I will follow that up with talking about fish now. And my project out here is identifying drivers of food web stability and fisheries productivity, specifically in Jackson Lake, and then using this as kind of a baseline to compare to other fisheries that we have around the state. And so kind of the question that's driving this is what energy pathways are supporting Wyoming reservoir food webs, including that of Jackson Lake, and how will those reservoir food webs respond to changing climates and these dynamic water levels that we've kind of been talking about to start this talk. And so a quick overview here of the Jackson Lake food web. We've been working with the Wyoming Game and Fish Department to understand specifically competition and predation between lake trout, brown trout, and Snake River cutthroat. So this is kind of how this project started. These are the 3 apex predators, or the top dogs of this reservoir. We have our lake trout, brown trout, and Snake River cutthroat trout here. It's a very popular lake trout fishery, and cutthroat, of course, are prominent throughout this area. And so how we're doing this work and quantifying these food webs is we're using stable isotopes. And so the real quick and dirty overview of this is we can use carbon, nitrogen, and sulfur that exist in different isotopic concentrations in the ecosystem to trace that flow of energy as small fish eat bugs, the bugs are eating the algae, and then those big fish are eating the small fish. And so we can use these isotopes to piece this all together. And this figure on the left here is the data that we get, and it's representing this pretty classic food web example on the right side here. And the 3 big fish on the top there, that's what we're really focusing on, is how many of these cutthroat and brown trout are the lake trout eating, and what sort of competition is occurring between these cutthroat and brown trout. Because the cutthroat, of course, are the native species in this lake, and brown trout are introduced. They're a pretty popular sport fish, but we want to make sure that these cutthroat can persist. And so some of these preliminary results that we're seeing here, we see a high overlap between these brown trout and Snake River cutthroat trout. And so this figure on the right here, basically each shaded circle represents a different species. And so this big blue circle is our lake trout. We can see that they have this really wide niche. So they're eating a lot of different things within the ecosystem. And then this yellowish circle and red circle are our cutthroat and brown trout respectively. And we see that they're overlapping quite a bit, which means that these species are eating a lot of the same food. And so when we kind of piece this together in the context of the rest of the ecosystem, we see that both these brown trout and Snake River cutthroat eat a lot of benthic resources. So this means they're eating bugs and stuff like that off of the bottom. And in this system, given that both species still appear to be doing really well, we expect that they're not resource limited. So there's enough food to go around. So even though they're eating the same thing, they're not stressing each other out. We also see that that big lake trout circle means that lake trout are escaping that competition with the other two species because of that broad resource use. So they're feeding higher on the trophic scale, so they're eating more fish and just a general wider pattern, so they can use the zooplankton out in the middle of the lake and some of those bugs in the nearshore habitat that the other species are eating too. We can also see that lake trout appear to consume more brown trout than cutthroat trout, which if you're into cutthroat persisting is a good thing. But across the board here, we see that most of the prey for these different species, especially our cutthroat and brown trout, are those benthic inverts, some Utah chubs, dace, other small-bodied fish and minnows that are out here in the reservoir. And also mountain whitefish are a big prey source for the lake trout out here, which makes sense because those lake trout and mountain whitefish both like to occupy that cold, deep water habitat out in the middle of the system. Another really cool thing that we can do with this isotope data is we can look at individual fish and how they're foraging. And so we can see that some of our lake trout are really specialized. We have some fish in these green boxes that are really keying in on mountain whitefish, just staying out in the middle. We can also see that we have some more generalist fish. So these might be the ones that are going to overlap with our brown trout and cutthroat, either through competition or predation. And so we can run these models for all of our different fish that we sample, and we can kind of see like, oh, some of this population is doing this thing, some are specializing, some are generalists. We can get some really cool results with this. And just to kind of wrap up some of this food web stuff for Jackson Lake here, with this plot of our isotope data, we can see that the food web is pretty balanced, which is a really good thing. And so this black arc through the middle of this plot is the total shape of the food web. And what we want is the peak of that arch to be right around 50% in the middle there, which means that half of the energy that's supporting these lake trout, the highest trophic level organism in the system, is coming from that pelagic offshore environment and this littoral nearshore environment that Sean and I have been talking about this whole time. What this means is that reductions in that cold water habitat volume could negatively affect those mountain whitefish that are a really important prey source for those lake trout. That could also have cascading effects, like we mentioned, when the water levels go down and we lose that littoral nearshore area, that's going to reduce the amount of those benthic resources that are available for the cutthroat and the brown trout. And so while they're coexisting peacefully now, in a scenario where water levels are chronically a lot lower, that could become a problem potentially. And to kind of carry this on, like across these different climate scenarios, we're hoping to continue to use this data to relate these changes in habitat availability to that competition between brown trout and Snake River and that predation on those two species by the lake trout. And then once we do that, we're hoping to apply these same methods to other water bodies across the state using Jackson Lake here as a baseline to compare those food webs to. And so, like I mentioned, we can look at the same data for reservoirs like Alcova or Pathfinder or Flaming Gorge, and we can look at the shapes of these systems and their food webs. And we can start to understand what sort of environmental drivers or fishery drivers might be affecting the shapes of these food webs. Also, like the specialization and generalization stuff that I talked about earlier with the lake trout out here, we can use this to look at the foraging ecology of these apex predators, primarily lake trout and walleye, across all of these reservoirs that we're collecting this stable isotope data for, to really understand what's driving this foraging behavior in these systems, and hopefully predict out what this might look like in the future. Another thing that we're working on right now is relating these environmental drivers to annual growth of these really economically and recreationally important species like walleye and lake trout. So we can age these fish using their ear bones or otoliths, and we can figure out how much they're growing each year, and we can pair that up with climate data and environmental data, and we can start to predict how they might grow in the future. Another thing that we're working on is the effects of stocking on predator populations. So as I'm sure a lot of you know, stocking is a major component of fisheries here in Wyoming. a lot of which is salmonids like rainbow trout or brown trout. And so we want to understand how that stocking is affecting these walleye and lake trout and things like that as well. And then finally, to wrap it up here, something that our technician is working on right now, which is super cool, is aging growth of Utah and white suckers. So this plot here is a growth, von Bertalant growth model of Utah suckers from the lake here. And this was pretty surprising to us when we first started doing this. A lot of the suckers, which are a commonly overlooked species, these fish are growing to be upwards of 60 years old. So they're lasting for a really long time, and they're growing pretty fast. And once they're out there and they escape that predation size, they're just kind of existing out there. And so we're hoping to expand this to a bunch of white sucker populations around the state and kind of incorporate this into the bigger picture of how this is influencing those food webs and those fisheries. And
okay, cool. Thanks everyone for sticking through this impromptu talk today. I'm Kevin. I'm a research tech on this broader WyACT project, and I lead a lot of the baseline data collection related to water quality across a couple different bodies of water in Wyoming that we'll talk about here. So I'm going to kind of take a step back and walk you through a lot of the work that we do, how we come to a lot of the data that the grad students are using. Kind of the research objectives for this kind of baseline water quality data is to start understanding these pretty data poor systems in Wyoming, at least in the grand scheme of what the data we work with as aquatic ecologists. There's not a lot of data for a lot of these ecosystems in Wyoming. And especially if we're going to make any statements about what's happening in the long term, we need to start collecting some baseline data. And hopefully we can provide this data to local managers and stakeholders within each of the watersheds that we work in, and then inform some more complex models of both present and future conditions for these aquatic ecosystems. So the three types of data collection that I'll talk about individually in my little portion of this talk are buoys, pretty self-explanatory, flame. I'll get more into that in a couple slides. And then some manual lake monitoring that we go out and do approximately every month throughout the summer. And here are our three primary lakes that we study, of course, Jackson Lake, but we also do a decent amount of work on Brooks Lake, just over Togety Pass, and then Boyson Reservoir down by Riverton, out east of the Tetons. And we collect a ton of different information on all these water bodies. The two parameters I'm going to focus on today are kind of foundational for water quality, are temperature and then the biomass of algae in these different lakes. And kind of just the question that we're hoping to answer here, and that hopefully you get out of this talk, are how do these three approaches tell us anything about these lakes, and how might they complement one another to give us a better holistic understanding of the ecology of these systems. So first I'll talk about buoys. As you've seen in a couple of these pictures, we have one out on Jackson Lake. We have them on Brooks and Boyson Reservoir as well. They sit out there from essentially ice on to ice off and record data every 10 minutes. So here's some of the high resolution data that we're getting from those. On the left, we have water temperature from 2023 and 2024, the last years that we've been doing this work. And then Boyson is at the top in purple, Jackson's in yellow, and Brooks is in this teal. That kind of makes sense because that tracks well with elevation. Of course, Jackson's in between, Brooks is high elevation up on Togety Pass, and then Boyson's down lower below the mountains. And then here's some time series of algae data. Jackson's down there in the yellow. You can see it's pretty minimal algae growth. That's what we expect. We don't see algal blooms out on Jackson Lake. But both Brooks Lake and Boyson do see algal blooms. So you can see these kind of peaks throughout the season. And that's when this really intense growth of algae is happening and causing what we call algae blooms. So, of course, here are pictures of the three lakes. The red stars are where buoys are sitting. But as you might imagine, they sit in one place all year. So they're not telling you everything about the entire surface of each of these lakes. We are missing information from all these different little inlets and bays of these different lakes. So how we collect some information across the entire surface of the lake is with a system that I mentioned earlier called FLAME. It's an acronym for Fast Limnological Automated Measurements. But it's basically a system where this kind of schematic on the right here is laying it out. You have a pipe that sticks down off your boat into the water, attached to some hoses and a pump and some water quality sensors. As you're driving around the lake, the pump pulls water in from the lake, runs it over your sensors and spits it back out, and records all these parameters on the left every second. So you get a pretty high resolution of the surface of the lake. And you can do this while you're driving. So we can drive across the surface of the lake in, anywhere from an hour to 3 hours and get a measurement of all these parameters every second. So what this data tends to look like then are these maps. And these colorful tracks are where we drove at different times of the year. At the top left is May, working our way through the seasons. To the bottom right is November. This is Jackson Lake, as you might recognize. So we're capturing the entire surface. Of course, we're not driving every single point of the entire surface, but we're getting a much more holistic understanding of surface water chemistry than we would if we just measured it at one point with a buoy. And so what these kind of figures are showing is that we see really strong seasonality, of course, in Jackson Lake. We see May and November, really cold water. These warmer colors represent warmer water up to upwards of 20 degrees C, which is above 70 Fahrenheit. But if you zoom into any one of these specific days, you'll see that there is variation across the surface at any one day you were out on Jackson Lake. So this is a day from last year. We were out on June 26th, and you can see that the colder colors are cooler temperatures. So you can see down there in the bottom left, Moran Bay is colder by a couple degrees, 3 to 4 degrees, than where the river comes in or down by the dam when the water's been sitting in the lake for a while. And we can do this on multiple days. So this is what it looked like in August. And you can see a little bit of a similar trend, but what sticks out here is that on the north, where the Snake River comes in, it's much colder because the river is much colder than a lake, which has been heating up in the sun all summer. And there's more variation than we might have expected in Jackson Lake. You can see in June, we had variation of almost 6 degrees, and then in August, around 3. So this is telling us that while seasonality is really important, there are also very localized environmental factors affecting temperature in Jackson Lake. And this is, these are just some bar graphs. On the left, we have temperature, on the right, we have chlorophyll. The height of the bar tells you what range of temperature or of algae we're seeing. So on the left, you can see temperature. The bars don't overlap very much because seasonality is such a strong driver there. On the right, this is algae. There's not a lot of algae at any time during the year, but those bars always overlap. So at any one moment you go out, you're going to see the same amount of variation in algae growth as you would if you went out in May versus November. Not the case for temperature. Pretty much what you'd expect there. And then I mentioned those other lakes that we study. I'll just touch on Boysen a little bit here. Here are some maps we made from Boysen. I guess the kind of comparison to Jackson here is that the temperature obviously gets a lot warmer down in Boysen. It's much lower elevation lake. And then they do have algal bloom problems, as I mentioned earlier. So you can see the scale of chlorophyll here is 0 to 5. The scale for the Jackson chlorophyll was 0 to 0.25. So Jackson has almost no algae, Boysen has a lot. And you can see that it's kind of hard to see on this graph, but the south end of Boysen and these small little bays tend to be the hot spots for their algal blooms down there, which we were able to detect with this sampling method. And similar to Jackson, we saw that within each day we were there. Here's June 27th of last year. There's a lot of variation across the reservoir down in Boysen, just like we saw in Jackson. We can see very patchy growth of algal blooms toward the south end, not up by the dam. And then in terms of temperature, we saw a 5 degrees C variation across the reservoir. And here's a similar plot to Jackson. You can see seasonality really matters for temperature. Spatial variation really seems to matter for chlorophyll. We saw that big algal bloom day in June, and much less as you got later in the year on Boysen. So we have our buoy in the red point. We have our flame track in the blue. So we got a pretty good comprehensive look at the surface water chemistry of Jackson. But what might we still be missing? Of course, in lakes, depth is really important. So all the data I've talked about so far are measured at the surface, but what might we be missing if we ignore what's happening below the surface? And that's where we work in some routine monitoring. So we go out every month on Jackson Lake here, we sample each of these yellow points and the red point, and collect a whole suite of the same parameters I've been talking about at different depths as we move down the water column. And one of the important things that we've learned from this is that our productivity or our algal growth actually doesn't peak at the surface. The surface is not where most of the algae are hanging out in Jackson Lake. It peaks at anywhere from 5 to 13 feet or so, 15 to 30 feet-ish. And you can see that these are lines are colored by year, so the darker colors are earlier in the year, and the warmer colors are later. And you can see that in June, these purple lines, we've had our largest peaks. They get the farthest to the right. And then as you move into September in the yellow, it's pretty straight up and down with not a lot of algae growth throughout any. And we think this is fueled by the Snake River runoff flows earlier in the year in May and June. So to wrap up my section of this talk, we've integrated all three of these kind of data collection approaches that I've talked about so far. We have our buoys, we have our flame that maps surface water chemistry, and then we have our manual monitoring that measures different depths. We've established a pretty comprehensive understanding of how the aquatic ecosystems in Wyoming are structured, and how this might then work its way up to all the biology, fish, the algae that we care about. And then hopefully this will feed future work, modeling both present and future conditions. And we are continuing to expand our data collection efforts. We had a question earlier about nitrogen or phosphorus limitation. We're actively trying to answer that question this summer. So more data are constantly coming in.
Hello, everybody. Thanks for being here. And you guys have heard a lot about the lake, but today I'll talk to you a little bit about what we're doing on the Snake River below Jackson Lake Dam. My name is Chuck Williams. I am currently a master's student with Willie Fetzer and Annika Walters. And my project is working on quantifying the thermal heterogeneity of the upper Snake River in Wyoming. That includes both cost, space, and time. So a little bit jargony there, but I'll kind of break it down for you. And so basically across this whole river here, from the dam all the way down to Moose, we want to quantify what's happening or put numbers to what's happening to the water temperature across this whole space and then seasonally as well. And so why is this important? So water temperature is considered a master variable controlled many physical, biological, and chemical processes within the river. This is especially important for local species like Snake River cutthroat. It can affect their metabolism, their species distribution, their growth. And this is important for this very culturally, economically, and ecologically important species. Supports a lot of fly fishing in the community, brings a lot of money. And so it's very important how these fish interact in this river with the temperature that they are a part of. And so management decisions are a lot of times based on single fixed site temperature collection. And that doesn't necessarily span the entire width of the river. And so part of what we're doing is using these fixed sites as well as measuring temperature along these flow paths to create like a snapshot of water temperature along the entire Snake River. So once we have this snapshot of the pitch of the water temperature of the snake, we need to look at what processes drive it and how those might change over time. So we might look at tributary inputs, maybe as areas of thermal refuge. We might look at what's happening with our climate, if it's warming or not, and then how that might affect water temperature across both space along the river and across time and season. So in our first objective, just to identify what is this snapshot, what are these patterns of water temperature? This is just a conceptual diagram. And for you, those of you who are here to see a volcano talk, I'm sorry, but it's about as close as we can make it. So we expect to see temporally, so across the season, we expect to see a little bit more heterogeneity in these lower water months in the summer. These are also really important months for trout growth and production. And then spatially, we might see, as we go along the river, we might see an increase in temperature and then a decrease near these tributaries. So we might see some heterogeneity or some changes in water temperature as we look at this spatially. The second part, looking at what processes or what drives water temperature. So near Jackson Lake Dam, the dam output is going to drive water temperature more. So then in the middle here or next to one of these tributaries, or how these climatic variables will potentially shift water temperature into the future. So just real quickly, we've already talked about this, but we are working from Jackson Lake Dam down to Moose, it's all within Grand Teton National Park. So we're seeing relatively little landscape landscape changes anymore. And so what we're doing in this section of river, about 50 kilometers long, is we are collecting two types of data to set up our patterns. The first is these fixed sites, so these little red dots, and we are adding a lot more. So there was originally one here at Jackson Lake Dam and one at Moose. We added a lot more along the way, and we're focused specifically on tributary inputs to where these changes in temperature might happen. So this is the Buffalo Fork coming in here, and we have set fixed sites above it, one above it, and then 3 below it. And this is a temperature we took while floating, and you can see how the Buffalo Fork decreases temperature for a certain amount of way below it. I'll get more into that later. So part of this is setting up those temperature loggers. So we use a wire connected to a piece of rebar tree with our temperature logger in there. Sometimes they get stuck, as you can see here. This is at Spread Creek. We eventually did get that one out. And sometimes we see a lot of people playing with them. So like we have some below Pacific Creek where popular boat launches, and I've walked up on kids just swinging my temp logger in the so anomalies in water temperature for sure happen. The other part is what we use is what Kevin talked about was this flame. So instead of having it on a boat and water pumped into the boat, we just put it in a PVC tube right here and float down the river. We have batteries contained within our dry box in our raft, and we float down and we collect water temperature. We'll also use a variable suite of landscape, and climate covariates to put into our model to help kind of look at what drives water temperature. You guys don't worry about all this as a so kind of our two questions, like how we're going to answer these with the data analysis. The first one, to create a snapshot of time, we are looking at using generalized additive models. So basically you put all your data into these and you train the model to predict what water temperature will be, say at 7:17 at night on July 24th. So then I'll create a snapshot of that image. The other model we're using is a stream spatial network model. It's called an SSN, and basically uses spatial regression to identify what covariates that were in that last graph are causing the water temperature to be what it is. So in this simulated data set, air temperature and stream temperature on a 1-to-1 ratio. So air temperature would be like the only thing affecting stream temperature, which we do not believe is true. And so this is all kind of like in progress. So I don't have any data to show you or any final results, but I would like to thank all these people here, because we've been working quite a bit with a lot of the community and stuff on this project. So